I think this talk is going to be a bit of an outlier for today, at least, in that there will be more machine learning and more biology, but I'm comfortable being an outlier, so we'll just go for it and see how it goes. Okay, so if you don't know my lab, what we do is we use single-cell data, spatial transcriptomics data, and 3D genomics data to try to understand gene regulation, but also kind of cellular ecosystems in tissues and tumors. And we do a lot of work developing machine learning models for regulatory genomics. But we also, with wonderful collaborators, do a lot of basic and cancer immunology and also cancer epigenetics. Okay, so today I'm going to focus on algorithms for understanding gene regulation. I'll motivate it a little bit and kind of give you an outline of what we'll cover. If there are a lot of questions, we should just deal with the questions and I can kind of lop off one of the topics, all right? So I'm quite flexible. Okay, so just a picture that hopefully people have in their mind of how gene regulation works in mammalian cells, okay? So you have distinguished elements, regulatory elements, that are the control elements for controlling expression of genes in a cell type specific manner, right? And we're familiar with promoters that are right close to the gene, right? But there are also regulatory elements that could be a megabase or more away in the linear genome from the gene, but are looping dynamically in 3D and making contact with the promoter. These elements are bound by DNA binding proteins called transcription factors. They recognize sequence-specific sites. We usually represent these sites that they recognize as these kind of motifs or logos, all right? So we have a code that we are trying to understand for decoding gene regulation. And our field is really driven by technology. And what has been exciting is we have new methods for mapping where these distinguished elements are. Okay, so the binding of transcription factors is holding open the chromatin, and we can map these regions of open chromatin with assays like ATAC-seq, and now we can do ATAC-seq in single cells. Okay, so single cell attack. All right. And the other thing that's been happening over the past 5 or so years is that 3D genomic technologies for mapping these 3D contacts, including enhancer-promoter interactions, have improved a lot. All right, so we can create these contact maps where we can see 3D architecture, physical proximity, contacts between enhancers and promoters, and that can also help us decode where are the regulatory elements, which ones are making contact with the promoter, what might be regulating genes, okay? So I'm going to focus on showing you how you can exploit new assays like single-cell chromatin accessibility, single-cell attack, single-cell multiome, where we have chromatin accessibility and the transcriptome in the same individual nuclei, and also 3D genomics to sort of decode gene regulation. Okay, so why do we care about gene regulation? So since this is a broad audience, you know, why does this help us, and why are people interested in these machine learning models of DNA sequence, of gene regulation? Well, one motivation is from cell engineering. Okay, so we have cell-based therapies. Very famously, we have CAR-Ts, where we're taking T cells out of a patient, engineering them to express a chimeric antigen receptor that is specific to, for example, a target on lymphomas cells, right? And then we infuse those engineered cells back into the patient. Or, you know, pancreatic beta cell replacement therapy. Okay, so we have these cell-based therapies, and now people are thinking about how do we control expression of a transgene and maybe synthetically design enhancers for the transgene. And this is a paper I just pulled out, it's from Stein Artz's lab, where they're using machine learning model to design an enhancer for a transgene, both in Drosophila and in human cell lines, and you can see it's expressed kind of in, I think that's the brain of the fly. Okay, so they're presenting this more as an experimental tool, but you can see sort of therapeutic applications coming. Okay. Another motivation that maybe people have already talked about is if you take the GWAS catalog, and we have, I guess this is saying almost 800,000 variant trait associations, right? And we're trying to interpret what these genetic variants do and in what cellular context they matter. So most of these genetic variants are non-coding, right? And they're not in LD with any coding region, right? And the coding exons in the human genome are 2% of the genome. And, but they are enriched in these chromatin accessible regions like enhancers and promoters where DNA binding factors are sitting, okay? So the hypothesis that is widely accepted is that, widely believed, what many people are kind of betting on is that these genetic variants, these non-coding genetic variants, are altering regulation of gene expression, all right? And, here's a picture, right? You have the underlying sequence of an enhancer or promoter, and maybe you have a single nucleotide polymorphism in the enhancer or promoter region, and that is altering the affinity of a transcription factor for binding, and that will change whether the gene is turned on or how much it is turned on in some cellular context that is disease relevant, okay? The picture, I don't know, is quite accurate. So it's sort of saying maybe you have a motif that the transcription factor recognizes and you ablate the motif with the genetic variant. I don't think it's always that easy. So often the variants are sort of more in the flanking regions. So it's more subtle. I think it's more subtle. But this is what the field is thinking about. Okay. And the other thing maybe people saw about 2 weeks ago, that big AI is now getting into regulatory genomics. So DeepMind just released AlphaGenome as a preprint. And this is a model that is trained to take a megabase of genomic DNA. It's trained on human and mouse. And it predicts thousands of epigenomic and transcriptomic tracks at base pair resolution. Also, some histone marks and 3D contact maps at not base pair resolution, a little more coarse-grained, okay? And in terms of architecture, it's sort of a scaled-up version of the Borzoi model from David Kelly's group, okay? But it is based on their published or not published, but reported benchmarking. It seems to be giving improved performance for variant effect prediction. Okay, so you can use these big models, you can put in the reference genome, you can put in the alternative, you make some predictions about how it would affect chromatin accessibility or transcription factor binding or splicing, okay? So, that's on its way. Okay, so what I'd like to talk about, 3 maybe less big models, more lightweight models, deep learning models to understand gene regulation. And first, I can quickly talk about a model called ChromaFold that was published last year. It's for predicting the high-C contact map from single-cell attack alone. Okay. Then I can talk about using 3D genomics information in predictive models of gene regulation. So using high-C, or it's actually going to be high-CAR data and chromatin accessibility in these models to predict gene expression, and then we can figure out what enhancers are regulating what genes. Okay, and if there's time, and I'll be rushing at this point, if there's time, I'm going to talk about another model where we use single-cell multiome data, so chromatin accessibility and RNA from the same cells, in a neural ODE approach to model dynamics of cells, and we're going to apply it to the germinal center reaction. Okay, so I like the model a lot, but it's probably the most complicated thing I'm going to talk about, and it's going to be at the end. Okay, so predicting 3D architecture to learn enhancer promoter links. Okay, so just if you haven't seen this before, we have these assays. I'm actually taking the figure from the ARIMA kit, all right? I have no relationship with ARIMA at the company, but just to show you, there are commercial solutions for generating high-C now, and I like the picture, all right? So the idea is you're cross-linking DNA in the nucleus. You have a restriction enzyme digest with the kind of cocktail of restriction enzymes. You ligate the ends together, pull down, and you get these kind of chimeric reads that are gray and blue in the picture, and you do paired end sequencing. And the idea is the gray and blue chimeric reads, maybe the gray part and the blue part, are far apart in the linear genome, but they came together, they were captured by this assay, so there was a contact in a cell in the input population. And that paired end read, we talk about the read pairs as anchors. We map them to bins in the genome, and we can say there's a contact between bin I and bin J, and produce a contact map, just as a matrix. All right. So this contact map, okay, oops. I think some of the figures are so big that there's a latency. All right, but the other thing you need to know, you can take this contact matrix, and you can either look at it as a matrix, or you can put it on the diagonal, so you can line it up with the genome and other genome tracks. So this is the diagonal version. Usually you normalize just with matrix rebalancing, so rows and columns summing to 1. And then you see these structures called topologically associating domains, these triangles. So regions of about a megabase that are interacting with themselves more than they're interacting with nearby regions. We want more information. So we actually have a model where we're estimating a background model directly from the data using negative binomial regression, and then we can assign a significance to the counts that we see that are bigger than we expect, okay? And these are z-scores from this method called HiCDC+. And you can see once you kind of assign significance to the contact bins and connect bins that are sort of significantly in contact, you see more structure, okay? And in particular, this is the MYC locus. MYC is an important oncogene. It's famous for having enhancers that are 2 megabases away. You can see them very clearly. You can actually see other structures. But this is the kind of information we're trying to get out of high-C. Who is talking to whom, and in particular, what enhancers are regulating what genes, what promoters. Okay, so high-C is great, but you still need a lot of input material. You need a complex library, and you need a lot of sequencing. So for high-C, to get a 5 kb resolution, typically you would want 1 billion or 2 billion reads, all right? And it might not even be feasible in smaller, rarer cell types, okay? So what is feasible with small cell numbers and looking at rarer cell types is single-cell attack, single-cell chromatin accessibility. Okay, and there's an idea from a paper, it's a very popular paper, a method called Cicero from some time ago. And their intuition was that maybe if you look at chromatin accessibility and you have 2 accessible events, like an enhancer and a promoter, right? And if they are opening and closing in a correlated fashion across cells or aggregated cells, metacells, then maybe they're more likely to interact in 3D, okay? So the idea is let's use a version of the co-accessibility matrix and just do a statistical processing of that co-accessibility matrix and predict interactions, okay? And they're just applying a graphical lasso approach to the co-accessibility matrix. Okay, is this gonna work? So I'm showing you in the same cell type, the co-accessibility matrix from single cell attack at the top, and this is the Hi-C contact map processed by our method at the bottom. And you can see there's a signal, right? Even, maybe this kind of co-accessibility you can sort of see here. There's a signal, right? But it's actually a pretty weak signal, all right? So you can imagine if you're just taking this co-accessibility matrix and applying a graphical lasso, it's not gonna work that great, all right? Another way to look at it is if you take true interactions and look at their distribution of co-accessibility in orange versus all interactions in the matrix in blue, you can see the orange is shifted to the right, okay? So there's a signal, but it's a subtle signal. All right, what can you do if you have a subtle signal? You can use a supervised approach with deep learning, right? Which is very good at taking subtle signals and getting accurate prediction, all right? So this is what we do. We take single cell attack. We do a co-accessibility matrix, just like that previous method, Cicero. But we also sample cells and generate pseudobulk 1D attack, okay? And that's here, okay? And we do different samplings to kind of model, to generalize better. We need a bit of sequence information. And what's important for getting TAT structure correct are CTCF binding sites, these kind of structural elements. So we just take a motif track for CTCF. It's free, right? So that's all we need. We don't have a full sequence model. We have 2 feature extractors. This one, after learning some representations, has an outer kind of tensor. And then we combine them and we predict these V stripes. Okay, so along the genome, every 10 kb, all the bins, this bin is interacting with forward and reverse in the genome up to 2 megabases away. Okay, and then we glue the V's together. Okay, how well does it work? So the first thing is just a sanity check. If you train within the same cell type and you hold out some chromosomes, how well does it generalize to the held out chromosomes, right? So this is not a real prediction task. This is just a sanity check to see you can actually learn something, okay? So within the training cell types, it actually looks pretty good, right? So this is a held out chromosome. This is the ground truth. This is the ChromaFold prediction. And the co-accessibility matrix, motif track, and pseudobulk attack. The CTCF chip we don't use, but just for reference, okay? And you can even see that we can make predictions through these regions where there are all these mappability artifacts in the Hi-C. Okay, so looks reasonable. That's not a real task. What we really want to do is generalize to new cell types or even new genomes. Okay, so here's we take the model that's trained on paired single-cell attack in high C and human ENCODE cell lines, and we apply it to mouse cell types. So new genome, new cell types. And again, I'm showing you, so all we have to make the prediction is single-cell attack in these mouse cell types, in mouse germinal center B cells, as well as regulatory T cells. And we happen to have the high C data, and this is all from wonderful collaborators, the Rudensky and Melnick labs. And you can see the ground truth and the prediction. It's not bad, okay? And it's actually good enough that, so this is the BCL6 locus, this is IKZF2 locus. These are important genes in these cell types. You might want to know what are the enhancers. And I'm also showing you the ground truth and predicted kind of interactions as loops. It's good enough that you could take these predictions and design CRISPR-I experiments and test the enhancers, and our collaborators are doing that, okay? So one more thing about this. All right, suppose you have a complex tissue like pancreatic islets that have different major cell types, and you have bulk high C that's representing sort of the population, level interactions, but you want to know which interactions come from which cell types. So what you can do is take this model trained on ENCODE cell line data, fine-tune on your single cell attack and high C data in your complex tissue, and then take that fine-tuned model and make predictions in each cell type, for example, in alpha cells and beta cells. Okay? And so that's what I'm showing you here. For a long time, we didn't know how to validate these predictions, but then someone actually published, sorted beta cells and alpha cells, and did a high C contact map, and we could assess that the predictions were not bad. Okay, not perfect, but for example, if you look at the glucagon locus, there's an enhancer that we predict only in alpha cells, not in beta cells, okay, this enhancer. And similarly, you can't see because I can barely see, but there are some beta cell specific enhancers at the insulin locus. Okay? Okay, so that's my first. This is published. I think it can work pretty well, right? So try it if you have single cell attack. Okay, so learning models of gene regulation from 1D and 3D genomics. Okay, so continuing on the theme of pancreatic differentiation. So this is a project with Danwei Huangfu at Sloan Kettering and Effie Apostolou at Weill Cornell. This is our 4DN project. And the idea is, let's look at pancreatic differentiation using guided differentiation of human pluripotent stem cells. Okay, so Danwei can take human ES cells and differentiate in a sort of multi-step protocol and eventually get these beta cells that secrete insulin. This is what's being used in replacement therapy for type 1 diabetes patients in clinical trials. The idea is we generated a high C-like data. It's called high CAR. It actually uses both transposase and restriction enzymes. It's from Yari Diao's lab, this protocol. And we also have multiome at these different steps of differentiation. Okay, so that will give us single cell expression and single cell chromatin accessibility. And can we link enhancers to genes? Can we interpret the cell type specific effects of type 2 diabetes, fine map variants? And can we build predictive models of gene regulation from this data? Okay, and here's just an example. All right, so this is high CAR data, and it's different stages of differentiation. So this is human ES cells, definitive endoderm, and pancreatic progenitor 1. Okay, and what we're looking at is the PDX1 gene. Okay, so it's an important pancreatic developmental transcription factor. It's a diabetes gene, And what you can see is these enhancer-promoter interactions are kind of gradually getting set up in differentiation. Okay, and I'm circling them in yellow, but you can see at PP1 that it's kind of this enhancer rewiring is happening, and now it's sort of set up to express that gene. Okay, and conceptually, maybe some of these diabetes variants are not affecting the fully mature cell types, but have some effect in differentiation. It's possible. Okay. Okay, so let me go quickly to show, there's a lot of cool things you can do just with the multiome data. All right, so what we have, this is a 7-step protocol, many days in between the different steps, especially towards the end when you want to get the late beta cells, late alpha cells. And we have 5 of the stages, okay? And I'm showing a UMAP of the RNA readout and a UMAP of the attack readout. Remember, we have parallel data, so they're coming from the same cells. I think what's cool is that even though the stages are separated by days, towards the end, you can start seeing some interesting structure, okay? And in particular, there's this sort of neurog3 or neurog3 positive cells that appear earlier, right, at the step 4, but then cluster with the step 5 and 6 cells, and you can start seeing maybe a trajectory from these progenitor cells into later cell types. And this embedding is actually a Kmer-based embedding, so it's a sequence-informed embedding of single-cell attack. I think I might have talked about it last year, but you can look it up. It's called cell space. Okay, so Kmers, using Kmers in accessible sites. Okay, so I want to go to how can we use this 3D information we have to link the connectivity and accessibility along the genome to gene expression. Okay, so a few years ago, we developed this method called GraphRig, okay? And it uses graph attention networks on a graph defined by high C edges, okay? So roughly speaking, when we get to the graph attention part, we think of the genome as kind of, the vertices are these 5 kb bins, and the edges are interactions defined by high C between 5 kb bins. So 5 kb is the resolution of the high C data, okay? There's 2 flavors of GraphRig. One takes epigenomic inputs, so some histone marks, and chromatin accessibility. We were using DNase instead of attack. And the output is CAGE. Okay, so CAGE maps the usage or the expression is a 5' protocol, so it tells you which transcription start sites are being used and kind of quantifies the expression of these at the 5' end. Okay, so first version, we have these. The context is really big, okay? So we're taking 6 megabases of context. It's still the biggest model, okay? That context is actually bigger than alpha genome. We have these edges that are of high C that are up to 2 megabases, okay? So the first step is we use just convolutional neural networks to learn features, representations of the epigenomic data. We pass that to a graph attention network so that we can pass information over these interactions between promoters and enhancers, and then we predict along the genome 5 kb resolution over the middle 2 kb. Okay, so we predict kind of this CAGE track. Okay, so that's version 1. Version 2 is we take 6 megabases of one hot encoded DNA sequence. We use the epigenomic tracks as an auxiliary task. Okay, so we have convolutional neural networks and dilated convolutional layers, and we're predicting the 1D histone marks and chromatin accessibility. So the idea is we're trying to learn motif-like information, motif-like representations that will predict these 1D tracks. We take this representation, give it to the graph attention network, pass information over these edges, and we predict cage again. Okay. And what's cool is you can use feature attribution on these models and figure out which enhancers are talking to which promoters. Even get some sequence information of what regions in an enhancer are important. Okay, and the one picture, so this is all published, the one picture I like from the paper is going back to the MIC locus. Okay, so as we know, MIC has these enhancers that are 2 megabases away. All right, so what I'm showing you is sequence-based model, or sorry, the epigenomic models and sequence-based models. And I'm using GraphReg versus a dilated CNN, which was the state of the art at the time. Okay, and I'm showing the prediction, but the important track is actually the feature attribution. So we're trying to explain the expression of MIC, and we're trying to show what are the regions in the genomic context that the models think are important for predicting MIC expression. And what you can see is GraphReg, because we have access to this 3D information, can figure out that these enhancers are important 2 megabases away. But the dilated CNN, the information just decays, right? So in principle, the receptive field is big enough that the model can see all this context, but only the promoter proximal information is really captured. And this is still true in the latest and greatest models, that the promoter information is better captured than more distal enhancers. Okay, the high C information really helps you here. Okay, so back to pancreatic differentiation. We have high CAR data, very nice, at these different stages of differentiation. We have multiome data that we can pseudo-bulk to represent different cell types at different stages. But that's it. We have chromatin accessibility and we have gene expression. Okay, so we don't have everything we had before. Okay, so the first problem is the RNA readout is a 3' capture. So we have expression at the gene level. We don't have this kind of 5' transcription start site information. All right. So the solution is that we kind of extend the graph, and we have nodes for genes that we see in the context, and we connect the gene to the bins that contain its annotated transcription start sites, and we have this kind of, extend the graph attention. So we're going to supervise at the gene level rather than at the bin level, which we could do before when we had cage. Okay, so that's the first thing. The second thing is that we only have a TAC-seq. We don't have these other histone marks. And a TAC-seq alone as an input is not as rich an information source, not as rich a representation. So what we do, and this is a postdoc, Yang Yang, who did all this, figured this out. She is first training a sequence model to predict chromatin accessibility. This is pseudobulk in a given cell type. And then she's taking a representation from that model together with the TAC-seq signal track and using that in a kind of GraphReg plus model. Okay, so 2 kind of tricks to extend. And again, just show quickly. All right, so I know these plots are crazy, right? But we're training a GraphReg plus model at every stage of pancreatic differentiation. Here we're looking at a gene called 1CUT1. The transcription start site is here. And we're doing feature attribution in red. Okay, so the red is saying what the model at each stage thinks is important for predicting expression of 1-cut-1. And you can see that there's an enhancer more than 600 kb away from the transcription start site that's getting attribution. You can see from, I know it's a bit messy, the high CAR, how it's figuring that out because there's a 3D link. This enhancer contains a type 2 diabetes risk variant. And we actually validated, we helped the Danwei Huangfu's lab validate this enhancer and how the variant changes actually the binding of a GATA factor. Okay, so an example of what you can do. This is another example going back to the PDX1 example that I showed you at the start. You can do these graph-reg models, and this is closer. This is an enhancer containing a type 2 diabetes risk variant. It's more than 400 kb from the transcription start site, and it's getting attribution. Okay, so you can use these models to kind of integrate different kinds of data, do feature attribution for specific genes, and then maybe annotate disease variants, fine mapped disease variants. Okay, how much time do I have left? Not clear.
You have, let's say 5 more minutes.
5 more minutes, okay. I'm gonna give you a very high level introduction to this model, but I think I can't tell you very much about it. Or maybe I should just stop and. Okay, I'll just introduce the idea because I don't have enough time to really show. Okay, so if you've worked with single cell data, you know there are a zillion trajectory inference methods. And the idea is that single cell sequencing is destructive, right? So you would love to watch the cells as they kind of go through a developmental process and trace them and see the actual developmental trajectory, but it's still really hard, okay? And certainly in vivo measurements with high plexity, very, very hard. So what we do is we can, take cells out of a mouse or out of a patient, and we can pretend that we're seeing a developmental process in a single time snapshot, that some cells are earlier and some cells are later, okay? And this is a method that I like from Donna Pear's lab called Palantir, and is kind of using a Markov process in a graph and sort of inferring these trajectories. Okay, so, all right. We're trying to do a neural ODE version of trajectory inference that is good for multiome. Okay, and where do we get the idea that we should have an ordinary differential equation anyway for learning dynamics? I mean, it's out there, and especially this RNA velocity idea is it's a very cool idea and widely used, though there are caveats that we'll get to, okay? So their idea is that from the actual count data, so this is RNA-seq only, you can kind of get spliced reads and unspliced reads. So reads from coding regions and reads from kind of the introns. And the intronic reads are sort of telling you about the pre-mRNA sort of less mature RNA, and the exonic reads are mature RNA. Okay, and this is all biologically actually not quite right. But then you can do a phase diagram of unspliced versus spliced, and when you turn on a gene, you see the unspliced reads first, and then when you turn off a gene, the spliced reads go down first. Anyway, you can write down some differential equations and solve one gene at a time. And if you have this for enough genes, you could get a vector field of rate of change of a bunch of genes, a rate of change for a set of genes, and plot this vector field and look at the flows and kind of get an idea of trajectories. All right. So problems is there's lack of robustness. There are a lot of parameters that can change your estimates. It can only be computed for a subset of genes. You don't have enough coverage. So it's kind of, there's some velocity genes. You get no information for the non-velocity genes. There's a lot of steps, okay? So you actually have to estimate a shared latent time from the individual gene models, okay? So there are things we want to get away from. So we're not actually using spliced and unspliced. What we're using is motif accessibility and gene expression from multiome, okay? So very quickly, we want to reduce dimensionality in a way that's still interpretable. So for every transcription factor, we can, if we have a motif, we can get a single cell estimate of the accessibility of its motif genome-wide, okay, using a method called ChromaFold. Okay, so it's kind of an inferred activity from accessibility of the motif, okay? So it's, you know, not at the level of the locus, but a kind of global readout of motif accessibility. And then we have gene expression, and we think that these two things, motif accessibility and gene expression, affect each other in a dynamic process that we're going to learn directly from the data, okay? So we are going to use neural ODEs, and in the 1 minute I have left, I'll just show you this picture. Maybe I can show you one data slide. We kind of have an encoder-decoder, so we map to a lower dimensional latent space. We learn the dynamics in the latent space, and then we go back to the observed space. What we're learning for every cell is its starting point in the latent space and the time, the latent time at which we you know, see the observable. And in neural ODEs, the neural network itself parametrizes the vector field, okay? And we use, you know, so I'm not writing down the loss function. We do a bunch of things, but one thing is we use RNA velocity on the velocity genes to supervise the dynamics, okay? So we use RNA velocity a little bit. Okay, so what's cool about this is that now you have a functional form of the dynamics, okay? So, you know, you can do pseudotime, you can do plots, you can sort of observe motif accessibility and gene expression over latent time, but you can also intervene in the system, right? You can do Jacobian analysis. What change of gene expression will affect the velocity of other genes. You can do an in silico perturbation of setting a transcription factor to its minimum expression and minimum accessibility and see how all the vector fields change. And you can even predict the effect of genetic loss of function. And if you have a genetic loss of function, you can test which perturbations would rescue and shift back to the homeostasis, okay? All of that is in the preprint. I'll just show you one thing, okay? So we apply this to germinal center B cells. They're very complex dynamics. This is the part of the antibody response. You know, the B cells somatically hypermutate their antigen receptor in the dark zone, transition to the light zone. They either get T cell help if it looks like the B cell receptor is doing well, or they die, right? They might go back to the dark zone, mutate some more, and eventually, you know, if they survive, they either exit as a plasmablast to make plasma cells that are just going to secrete antibodies, right? Or pre-memory B cells, they'll make memory B cells for the next infection. They'll be around, okay? So very important very complicated cycles, 2 exits, right? And we can learn these trajectories, and we can also do in silico perturbations and Jacobian analysis. And I'm just going to end with this one slide that, sorry that I should have maybe shown you this because it's the newest thing, but we can actually learn all the interactions that are known in the literature, that transcription factor, target interactions, just from this modeling. And then we also have data in an ARID1A heterozygous loss. Okay, so this is a mutation that you see in lymphomas and also a loss of CTCF. We can predict what interactions are lost when you have these genetic changes. Okay, so that's mainly what I wanted to get through. There's more details in the paper. Maybe next year I can give the whole talk on this, but let me just thank everybody who did the work. GraphReg and Dynavelo's Reza Karbalayghareh, Vian Gao did ChromaFold, and Zhan Li and Yang did the work on pancreatic differentiation.